Welcome back to the deep dive. Today we are undertaking a truly monumental journey into the foundational science that is actively transforming medicine.
It is. We are living squarely in what people are calling the genomic era.
And these huge revolutionary changes that are happening right now in healthcare, they're all being driven by this powerful new suite of technologies.
It's hard to overstate the magnitude of the shift. I mean, if you just look back 2 decades, the Human Genome Project sequencing, that first human blueprint, took 12 years.
12 years and what, something like $3 billion?
An estimated 3 billion, yes. And that was painstaking, targeted work, analyzing one single gene or just a small section at a time.
And now, what does that look like today?
Now, we can sequence an entire human genome, all 3.1 billion of those base pairs, in a single day, and for less than $1,000.
That is the very definition of a technological revolution.
Absolutely.
Oh.
We've moved from that slow, one-by-one analysis into this realm of parallel sequencing, where we're analyzing vast numbers of genes all at the same time.
Yeah. Okay, so let's unpack this revolution, because it's not just a story of faster, cheaper tech. It's really a story of fundamental biology becoming actionable.
That's the word for it.
So our mission for this deep dive is, it's comprehensive. We're going to start by really nailing down the fundamentals of genomics, the very blueprint of life, and all the intricate ways that information is regulated.
And then we'll get into how errors can arise, both the constitutional kind you inherit and the somatic kind you acquire.
Yeah.
Which are what cause disease.
Exactly. Then we'll dive deep into the core technologies that are powering all of this, from, the established techniques to the absolute bleeding edge of next-generation sequencing.
Right.
And finally, we'll look at how this explosion of data is actually being applied across critical medical specialties: obstetrics, pediatrics, oncology, even infectious disease. And critically, we have to review the profound ethical impacts these powerful tools have on individuals and their families.
It's a crucial mission because, as you mentioned, these tools are moving so rapidly out of that niche rare disease world and into directing therapies for very common conditions and providing crucial preventative information.
So understanding a person's genomic profile is becoming, a necessary component of just regular clinical care?
It is, yeah. To do it safely and effectively. This information, it's not just for the geneticists anymore. It's for everyone.
So let's start at the very beginning, the physical blueprint. When we talk about genes being these functional units of life, what exactly are they encoded in, and how is that massive amount of information physically stored in a cell that we can barely even see?
Okay, so genes are encoded in double-stranded deoxyribonucleic acid.
DNA.
Wow. And at its heart, it's a linear sequence made up of just 4 chemical bases.
Yeah.
Adenine, which we call A, cytosine C, thymine T, and guanine G.
And these bases form that universally recognized double helix, the twisted ladder structure we all learned about in school.
That's the one.
But what's the secret to its stability? What ensures the fidelity of that structure, and therefore the fidelity of the genetic information when a cell has to divide?
That comes down to a really crucial rule, the rule of complementarity.
The pairing.
Exactly, the pairing. The stability of that double helix relies on these weak hydrogen bonds holding the 2 strands together, but they only work if C pairs with G and A pairs with T.
And that's not just for structure, right? That pairing is functional.
It's the whole basis of function. It enables faithful DNA replication when the helix unzips, because each strand can serve as a perfect template. And it's also the very mechanism that allows our modern sequencing technologies to read that template.
Let's just take a second to appreciate the sheer scale here. You mentioned 3.1 billion base pairs in a single copy of the human genome.
Wow.
If we stretch that DNA out end to end, it would be about 6 feet long.
It's incredible.
So how on earth do you package 6 feet of material into a cell nucleus that is just a few microns wide?
The packaging is, it's an engineering marvel. It uses multiple layers of coiling. This massive DNA molecule is first wound very tightly around proteins called histones.
Okay, so you can think of the histones as, spools for the DNA thread.
That's a perfect analogy. And that brings us to the structure of the nucleosome and chromatin.
Right.
So the basic storage unit is called the nucleosome. That's about 147 base pairs of DNA wrapped around a core of 8 of these histone proteins.
And then those get packaged even further.
Yes. These nucleosomes then string together, and they look a bit like a string of beads, and then they're further wound and packaged into progressively thicker fibers to form what we call chromatin.
And what I find particularly amazing is this next point, that chromatin isn't just one uniform tangled mass. It actually exists in different states that physically determine whether a gene can even be switched on or not.
That's right. This physical configuration is absolutely vital for regulating gene expression. We can differentiate between two primary states. First, there's heterochromatin.
And that's the tightly packed stuff.
Exactly. It's tightly wound, densely packed, and generally it's transcriptionally silent. It's basically locked down and inaccessible.
And the opposite of that.
The opposite is euchromatin. This is open, it's less tightly wound, and it's physically accessible to all the transcription machinery. And so it's transcriptionally active.
So the cell is constantly changing this configuration.
Rapidly. It's making decisions about which genes it needs to express at any given moment by opening and closing these chromatin regions.
And finally, all of this coiled chromatin is packaged into the largest structures we can actually see under a microscope, the chromosomes.
Correct. Humans are diploid organisms, meaning we possess two copies of the genome, one from each parent, and microscopically this is visible as 23 pairs of chromosomes, which make up our karyotype.
So chromosomes 1 through 22 are the autosomes, and then that 23rd pair are the sex chromosomes, XX for female or XY for male.
That's right.
So once it's all packaged, the cell needs to actually access that information and express it. And it does that primarily by making proteins. This is what we call the central dogma: transcription, splicing, translation, and then protein modification.
It is, but what's fascinating is that while we still call it the dogma, we now know it's a huge simplification.
How so?
Well, the old idea of one gene equals one protein is long gone.
Mm-hmm.
We now know that a single gene can often encode many, many different proteins, which completely defies that simple linear relationship. But the dogma still gives us the necessary framework to understand how the information flows.
Okay, so let's focus first on transcription, getting from DNA to messenger RNA or mRNA. Who are the key players in that process?
The key player is an enzyme called RNA polymerase. This enzyme binds to a specific start sequence on the DNA, which we call the promoter region.
And it just reads the DNA.
It moves along one strand of the DNA template, and as it goes, it produces a complementary RNA molecule. For protein-coding genes, this is the mRNA. And then transcription ends when the polymerase hits a termination sequence, often called the polyadenylation signal.
And the RNA product that it makes is chemically distinct from the DNA template it was reading.
There are three critical differences, yes. RNA is generally single-stranded. It uses a different sugar ribose instead of deoxyribose. And this is key for sequencing. It contains a base called uracil, or U, in place of thymine, T.
So where the DNA strand has an A, the RNA incorporates a U.
Exactly, not a T.
Now, transcription isn't passive, it's highly regulated. This is where things called transcription factors come in, binding near the promoter. But here's where the regulation gets really fascinating, the enhancers. These can be, miles away from the gene itself?
It seems that way. Enhancer elements are regulatory sequences that can be located tens of thousands, sometimes hundreds of thousands of base pairs away from the promoter that they are controlling.
So how do they interact if they're so far apart?
The chromosomal DNA has to physically loop back on itself, bringing the enhancer and the proteins bound to it right next to the promoter to, modulate the RNA polymerase activity.
That concept of a physical loop controlling a gene that's so far away, it's hard to visualize, but it's incredibly powerful. Do we have a good clinical example of what happens when that distant regulation goes wrong?
A very clear one. Alpha thalassemia. It's a blood disorder.
Right.
And while it's often caused by deletions of the gene itself, it can also result from a variant in an enhancer element that's located over 100,000 base pairs away from the alpha-globin gene promoter.
So the gene itself is totally fine.
The gene is perfect, but the transcription is greatly reduced because that loop structure and the enhancer function are disrupted.
This leads us directly to epigenetics, the regulatory mechanisms that sit, as the name implies, above the genome itself. These are modifications that affect gene expression without changing the underlying DNA base sequence. What are the two main ways this happens?
So the first is DNA methylation. This involves adding a little chemical tag, a methyl group, to cytosine bases, usually in the promoter regions of genes.
And what does that do?
When a promoter is methylated, transcription factors can't bind effectively, and transcription is typically silenced.
So methylation is basically a silent switch for specific genes. What's the second mechanism?
The second mechanism involves the packaging itself, those histone tail modifications we touched on earlier. Chemical modifications, like methylation or acetylation, on the histone protein tails determine the configuration of the chromatin.
So it controls whether it's open or closed.
Precisely. Acetylation, for example, tends to loosen the coil, which promotes active chromatin, while certain types of methylation can promote that tight, inactive heterochromatin.
And what's fascinating here is that we now know that abnormal epigenetic changes are central to disease pathology, particularly in cancer.
Absolutely. Abnormal epigenetic changes, like widespread inappropriate methylation, are critical drivers in cancer. It allows the inappropriate expression of normally silenced genes, which can lead to uncontrolled proliferation.
But that also means it's a huge area for
A
therapeutics.
huge area.
Can you give us an example of an epigenetic therapy?
Certainly, there's a class of drugs called histone deacetylase inhibitors, like vorinostat.
Okay.
Vorinostat is used to treat cutaneous T-cell lymphoma. And what it does is it inhibits the removal of those acetyl groups from histones, which forces the chromatin to stay open in that euchromatin state.
So it's re-expressing genes that were silenced.
Exactly. It re-expresses genes that promote T-cell differentiation instead of tumor proliferation.
Yeah.
It effectively causes the tumor cells to mature and then regress.
So once transcription creates that primary RNA transcript, it's still not ready for protein production. It needs some serious editing. This is RNA splicing.
That's right. The primary transcript is huge. It contains copies of both the coding regions, which we call exons, and the non-coding regions, the introns. Splicing is the process that removes the introns and then retains and rejoins the exons,
The parts you actually need.
the segments necessary for creating the final functional protein blueprint.
And this process isn't fixed, is it? This is where alternative splicing comes in, which adds that layer of complexity you mentioned earlier, allowing one gene to produce multiple distinct proteins.
This is a major reason why the human genome only has about 20,000 protein-coding genes, yet we can generate vastly more proteins than that. Alternative splicing means we can choose different combinations of exons from the same primary transcript.
The calcitonin example you mentioned in our notes is a perfect illustration of this single gene dual function.
It is. In the thyroid C cells, the calcitonin gene produces an mRNA that encodes the hormone calcitonin, which regulates calcium. Simple enough.
But in neurons,
In neurons, the exact same gene is transcribed, but then alternative splicing occurs. And this yields an mRNA that encodes a totally different protein, a powerful neurotransmitter called calcitonin gene-related peptide.
same gene, different tissue, different splicing, totally different function. Incredible.
It's biological efficiency.
So once the mRNA is spliced and processed, it exits the nucleus and it serves as the template for translation, which is protein production. The code itself is read in these 3 base units, or codons, which define what's called the open reading frame.
Right, and we have 64 total codons. Here's a great example of biological efficiency and redundancy. 61 of those codons encode the 20 common amino acids.
So there's some overlap.
There's redundancy, exactly. Some amino acids are coded for by multiple codons. The remaining 3 codons, UAA, UAG, and UGA, are the stop codons. They terminate the polypeptide chain.
And what's the physical factory that executes this assembly line?
That's the ribosome. It's the site of protein production, and it's a complex molecular machine made up of non-coding ribosomal RNA, or rRNA, and proteins. The mRNA associates with the ribosome, and then we introduce the third key RNA type, which is transfer RNA, or tRNA.
And the tRNA is the translator.
It is. The tRNA molecules deliver the appropriate amino acids by matching their 3-nucleotide anticodon sequence to the complementary codon on the mRNA.
So once that amino acid chain is assembled, the protein is still not finished. It requires complex post-translational modification. Why is all this folding and processing so vital?
Because if the protein doesn't fold correctly, it can't function correctly,
Yeah.
or can't even be transported to where it needs to go.
And this happens in specific parts of the cell?
It often happens in the endoplasmic reticulum, the ER, and the Golgi apparatus. That's where the folding is refined, and modifications like glycosylation, the attachment of sugar molecules, take place.
And the most common cystic fibrosis variant provides a really stark illustration of what happens when this modification process gets disrupted.
It does. The most common CFTR variant, F508, results in a protein sequence that's just slightly abnormal. But even though the cell is perfectly capable of making the protein, the quality control checkpoints in the ER and Golgi recognize the folding defect.
So that they don't let it out.
They prevent the protein from being exported to the cell membrane, which is where it needs to be to function. And that failure of transport is what causes the disease symptoms.
Finally, we have to mention the surprising power of non-coding RNA, or ncRNA. For so long, we just focused on the protein-coding genes. But now we know this non-coding landscape is incredibly rich in regulatory power.
Yes, roughly 4,500 human genes encode functional ncRNAs like the tRNA and RNA we just mentioned. But the real regulatory stars here are the microRNAs, or miRNAs. We have over 1,000 different types.
And what's their role?
Their role is powerful post-translational gene expression control. They bind to target mRNAs, typically in a region called the 3' untranslated region, and they act like a tag that promotes the degradation of that mRNA.
So it effectively silences the gene product.
Exactly. It's an incredibly efficient way to shut down a gene product quickly. And their reach is astonishing. They are estimated to regulate over half of all human genes.
Wow.
They play huge, complex roles in development, in cancer progression, degenerative disorders. They're targets of intense therapeutic research right now. They're really the fine-tuning mechanism of the genome.
Having established the blueprint, we need to understand how cells replicate it, both for general growth and repair and for reproduction. Let's start with mitosis, which is the division process for our normal somatic cells.
Mitosis is a remarkable and very tightly controlled process. It ensures that two genetically identical diploid daughter cells are produced. Cells cycle through G1, which is growth, S phase, where DNA replication happens, and G2, which is for error checking, before they enter M phase, the actual division.
And what is the crucial element that prevents errors from accumulating during all of this?
The cell cycle checkpoints. The most critical one is the checkpoint between G2 and mitosis. This checkpoint's job is to ensure that every single piece of damaged DNA is repaired before the cell even attempts to divide.
And when those checkpoints fail, that's a direct path to pathology.
Yes. Failure of these checkpoints is a major driver of cancer pathogenesis. The cell loses its ability to repair variants, and it begins to rapidly accumulate more and more genomic damage, which leads to uncontrolled growth.
Okay, now for meiosis, the specialized division process for gametes, the sex cells. And this process is fundamentally different because it has to reduce the chromosome number.
Right. Meiosis involves two rounds of division, meiosis the first and meiosis the second. And it reduces the chromosome number from diploid, which is 46, to haploid, which is 23.
And that reduction is absolutely necessary.
It has to happen.
Yep.
So that when the egg and sperm combine, the resulting zygote returns to the normal 46 chromosome complement instead of having 92.
And it's also responsible for the huge diversity we see in offspring through recombination.
Recombination, or crossing over, is where homologous pairs of sister chromatids literally swap genetic material. This happens at the end of meiosis the first. And this ensures that the chromosomes you pass down to your offspring are mosaics, a mix of your own maternal and paternal chromosomes, which contributes significantly to genetic variation.
Here is a fascinating difference. The timing of meiosis between males and females is vastly different, and that timing is tied directly to age-related errors.
The timing discrepancy is immense. In males, meiotic division starts at puberty, and it's completed within days. In females, however, meiosis begins during fetal life.
In the womb.
In the womb.
Yeah.
And then it pauses in a prolonged stage until after ovulation. This means a single-cell division can potentially take over 40 years to complete in an older woman.
And the stress on that cellular machinery over decades must lead to errors.
Exactly. The reduced efficiency of chromosome separation in older eggs leads to something called non-disjunction, the failure of homologous chromosomes to separate properly.
And that's the cause of Down syndrome.
It's the direct cause of the increased risk of trisomies, like Down syndrome or trisomy 21, with increasing maternal age. The cellular machinery simply doesn't age as well when it's sitting paused for decades.
Even outside of meiosis, our cells can't divide forever. They have a built-in limit called the Hayflick limit, which leads to senescence.
The Hayflick limit just defines the limited capacity for division in most of our non-stem cells. Once cells hit this limit, they enter a state of senescence where division stops. And this natural process is intimately linked to the study of normal aging.
And studying diseases that accelerate this process can provide clues about the mechanisms.
Yes, these progeroid syndromes are rare diseases that dramatically accelerate aging, and they often highlight failures in DNA repair.
Like Werner syndrome.
Werner syndrome is a great example. It's caused by a mutation in a DNA helicase, which is an enzyme that's crucial for separating DNA strands during repair and replication. When it fails, DNA damage accumulates rapidly, leading to the symptoms of premature aging.
Now let's move from a limited life to active destruction apoptosis, or programmed cell death.
Apoptosis is essential. It's essential for development. I mean, you can think of the cells that carve out the spaces between your fingers and toes in the embryo, and for immune defense, it's a highly controlled, active process.
Triggered by specific signals.
Exactly. Those signals activate a family of enzymes called caspases. Caspases are the executioners. They actively destroy the cellular components, including the DNA.
And what makes this process so neat and clean and so crucial to tissue health?
The resulting fragments are packaged into these organized little structures called apoptotic bodies, which are then efficiently removed by phagocytic cells like macrophages. And crucially, this all happens without causing inflammation, so it preserves the integrity of the surrounding tissue.
Which contrasts sharply with necrosis.
Necrosis is pathological. It's uncontrolled cell death. It's usually caused by an external injury or a lack of essential components, most commonly hypoxia, a lack of oxygen.
So it's messy.
It's very messy. The cell swells and bursts, releasing its contents into the environment, and that is what triggers inflammation.
Okay, now we move to the absolute core material of genomics. Variation. We all acknowledge we carry millions of differences, most of which are totally benign. What defines those differences?
On average, each of us has somewhere between 3 and 5 million variants compared to the reference genome. That's about 1 every 300 base pairs. Most are just simple, common polymorphisms, which we define as occurring in more than 1% of the population, and they are usually single nucleotide polymorphisms, or SNPs.
SNPs.
SNPs, yeah.
Let's focus on the impact of those single base changes, nucleotide substitutions, especially the ones that fall within the open reading frame. The consequence really depends entirely on the resulting codon, doesn't it?
Entirely. We categorize the impact into 4 main types. First, synonymous.
That's a silent one.
It is. It changes the codon sequence, but doesn't change the amino acid that is encoded, so silent. Second is non-synonymous, or missense. This changes the codon and results in a different amino acid. The protein structure and function may or may not be affected.
And then the really severe one.
Stop gain or nonsense. This is generally the most severe. It introduces a premature stop codon, which results in a severely truncated, usually non-functional protein. And finally, you can have splicing variants, which typically occur at the junctions between introns and exons and disrupt normal RNA processing.
Then we have insertions and deletions, or indels, where the number of bases involved is absolutely key to determining the severity.
The rule of 3 is everything here. If the insertion or deletion involves a multiple of 3 bases, it's considered in frame and it maintains the reading frame. It might add or remove an amino acid, which can be disruptive, but the rest of the protein sequence downstream remains correct.
But if it's not a multiple of 3, say 1 or 2 nucleotides,
Then it causes a severe frame shift variant. This totally disrupts the sequence of all the downstream amino acids, and it almost always results in a premature stop codon shortly thereafter. That leads to a completely non-functional protein.
finally, we have the unstable repeat variants. These are variations in these simple tandem repeats that can actually expand across generations.
Right. These repeats, often called microsatellites or mini-satellites, they can arise due to DNA slippage during replication. The crucial point is that they are unstable. The longer the original repeat is, the greater the likelihood it will expand or contract during meiosis.
And these expansions are the cause of several well-known neurological diseases that display anticipation, meaning they get worse or start earlier in subsequent generations.
The classic examples are the triplet repeat expansion disorders. Huntington's disease is caused by a CAG expansion in the coding DNA,
Right.
and that leads to this elongated toxic polyglutamine tract in the protein. Conversely, Fragile X mental retardation is caused by a CGG expansion, but this occurs in the non-coding 5' UTR. And this large expansion leads to hypermethylation of the promoter, which silences the gene entirely.
Okay, so moving up in scale, we have copy number variations, or CNVs. These involve changes in the number of copies of large segments of the genome or even whole chromosomes.
The largest of these are the numerical anomalies, so whole chromosome gains or losses, which is called aneuploidy. We already mentioned they result from non-disjunction in meiosis.
So the common trisomies are 21, 18, and 13.
Right. And crucially, the only constitutional autosomal monosomy, that's the loss of one chromosome, that's compatible with life, is Turner syndrome, which is 45X, the monosomy of the X chromosome.
Then we have structural anomalies, so large insertions or deletions of chromosomal segments. These are almost always highly detrimental, right?
Almost always, yes.
Hmm.
They usually lead to severe congenital malformations or learning disabilities.
Yeah.
And these large structural changes often arise from errors in DNA repair mechanisms, like non-homologous end joining, where broken ends are just ligated together incorrectly.
We mentioned that array CGH, a technology we'll get to, helped us uncover a new class of smaller structural issues, the microscale anomalies. What differentiates these from the big structural anomalies we just discussed?
It's primarily their size. They're too small to detect by traditional light microscopy, by karyotyping, but they're easily picked up by the array technologies. And these contiguous gene defects often result in defined, recognizable syndromes.
So a single deletion or duplication can cause multiple distinct clinical features.
Exactly. A great example is DiGeorge syndrome, which results from a specific microdeletion on chromosome 22. Others include Williams syndrome, or the complex region on chromosome 15, associated with both Prader-Willi and Angelman syndromes. It just illustrates that the gain or loss of a tiny segment can have a profound impact on development.
Okay, so constitutional genetic diseases are the ones inherited through the germline. And before we even start talking about the gene, the clinical geneticist relies on the pedigree, the family tree, as their primary tool.
The pedigree is absolutely indispensable. You need a detailed, accurate three-generation history on both sides of the family. You need to know about medical conditions, developmental issues, any history of pregnancy loss. It provides the visual pattern that informs the initial diagnosis and helps you determine the pattern of inheritance.
Let's look at those patterns, starting with autosomal dominant or AD inheritance.
In AD inheritance, having just one mutated allele is enough to cause the condition. The key features are that you see affected individuals in every generation, unless the variant arose de novo or new in that affected person.
And it affects males and females equally.
Equally, and the offspring risk for an affected parent is 50% for each pregnancy. Lynch syndrome, which predisposes people to colorectal and endometrial cancers, is a classic AD condition.
But the clinical picture in these AD conditions is rarely so perfect, and that leads us to two key complicating concepts: penetrance and expressivity.
Yes, so penetrance is the proportion of people who carry the pathogenic variant and actually develop the disease phenotype. If the penetrance is low, say 80%, it means 20% of the carriers are walking around perfectly healthy, which can make tracing that pedigree very difficult.
It looks like it's skipping a generation.
It does. And then expressivity describes the severity level. An AD condition can manifest very mildly in one family member, that's low expressivity, and very severely in another, high expressivity.
Okay, next up, autosomal recessive, or AR inheritance. Here, both alleles must be mutated for the disease to manifest, meaning the parents are usually healthy carriers.
The pedigree features here are very distinct. Again, equal gender split, but the condition often affects only one sibship. You see the disease skipping generations and then suddenly appearing out of the blue.
And the risk is highest when there is consanguinity.
Right, because that increases the likelihood of inheriting the same rare mutant allele from both parents. And the offspring risk for two carrier parents is 25% for each pregnancy. A great example of AR is alpha-1 antitrypsin deficiency, where the PZ phenotype results in severely reduced levels of the protein.
Moving on to X-linked inheritance. This is primarily recessive, and it predominantly affects males because they only have one X chromosome.
Right, and transmission is through unaffected females, as they can carry the variant without exhibiting the disease. Affected males cannot transmit the condition to their sons.
Because they pass on their Y chromosome.
Exactly. But all of their daughters must be carriers.
What about the rare cases where a female carrier of an X-linked recessive disorder does show signs of it?
That's usually due to something called skewed X inactivation. Early in female embryonic development, one of the two X chromosomes is randomly inactivated in each cell. If, just by chance, the normal X is disproportionately inactivated across tissues, skewing towards the mutant X being active, then the female carrier might display some mild or moderate signs of the disorder.
And Duchenne muscular dystrophy, or DMD, is a tragic example of this inheritance pattern.
It is. DMD is X-linked recessive. It involves large deletions or variants in the dystrophin gene. And because dystrophin is necessary for muscle structural integrity, its absence results in severe, progressive muscle wasting, typically leading to the death of the affected male in his mid-20s from respiratory failure.
Finally, we have to address mitochondrial inheritance, which is a totally different beast. It completely bypasses the nuclear inheritance patterns we just discussed.
It does. Mitochondrial DNA, or mtDNA, is small and circular, and it's transmitted only by the oocytes, the egg cells.
Because sperm mitochondria are degraded upon fertilization.
Right, so inheritance is exclusively maternal. Males and females are affected equally, but males cannot pass the condition on.
And the variability in clinical presentation, even within the same family, is due to this concept of heteroplasmy.
mtDNA heteroplasmy. It means that only a proportion of the mtDNA molecules within a given cell contain the causal variant. So the disease severity really depends on the ratio of mutant to normal mtDNA. And the tissues with the highest metabolic demands, muscle, retina, the basal ganglia of the brain, are the most severely affected.
Imprinting disorders add another layer of complexity where gene expression depends not just on the gene itself, but on which parent you inherited it from.
Right. Imprinting is defined as gene expression being inherited in a parent of origin specific manner. For specific chromosomal regions, the paternal copy of a gene might be active while the maternal copy is silenced through methylation, or vice versa.
So if the mutation occurs on the silenced allele, there's no consequence. But if it occurs on the active allele, the child essentially has 0 functional copies and the phenotype manifests.
Exactly. And this can create spectacular contrasting phenotypes from defects in the very same chromosomal region. The classic region is 15q11q13. So loss of the active paternal contribution causes Prader-Willi syndrome, which is characterized by childhood obesity and developmental delay. But if there is a loss of function in the maternally inherited UBE3A gene, which is predominantly expressed only in the brain, it causes Angelman syndrome.
Which is completely different.
Totally different, characterized by severe mental retardation, epilepsy, and ataxia.
So up until now, we've been discussing constitutional or inherited variants. But most of the common genetic diseases we face, particularly cancer, are driven by somatic variants which occur after conception.
Cancer is absolutely the most important somatic genetic disease. It results from an accumulation of what we call driver mutations in genes that regulate cell division or program cell death, leading to abnormal cell growth and tumor formation.
And we categorize these driver mutations into 2 primary types, often called the gas pedal and the brakes of the cell.
A great analogy. Oncogenes are the gas pedal. Variants here usually result in a gain of function, promoting uncontrolled growth. Think of RAS mutations. Tumor suppressor genes are the brakes. Variants here require a loss of function in both copies of the gene to lose control. Think of TP53 or BRCA1.
And most cancers are a result of both.
It's a cocktail, an accumulation of variants in both types of genes.
This concept of accumulated variants is famously formalized in the Knudsen hypothesis, or the 2-hit mechanism, which explains why familial cancer syndromes happen.
The hypothesis is just elegant. In familial cancer syndromes, the individual inherits a germline loss of function variant in a tumor suppressor gene that's hit 1.
But they're healthy at birth.
They're healthy because they have 1 remaining normal copy. Tumor development only begins when a somatic event hit 2 affects that remaining normal copy later in life. This complete loss of tumor suppressor activity is what triggers the cancer.
And that explains why someone with an inherited predisposition, like in BRCA1, might not develop breast cancer until their 40s or 50s.
Exactly. They're waiting for that second hit.
Okay, let's shift gears completely and look at how we actually measure and detect all these variants. The technological pace here is just astounding. Let's start with the traditional method, karyotyping.
Karyotyping was the mainstay for decades. It involves analyzing metaphase chromosomes under a light microscope. It could detect whole chromosome gains or losses, like trisomy 21, or very large segmental anomalies.
And by large, we're talking.
We are talking about structural changes greater than 4 million base pairs. But it was laborious, and it really lacked resolution.
And this has now been largely replaced by array comparative genomic hybridization, or array CGH. This jump in technology immediately increased our resolution.
It did. Array CGH uses fluorescence to achieve rapid and precise detection of segmental gains or losses across the entire genome, regardless of their size.
So how does it work?
You take patient DNA and you label it with, say, a green fluorescent dye, and you take reference DNA and label it red. Then you hybridize them to a microarray. If the signal deviates from that expected 1-to-1 yellow ratio, you can see a deletion, which would be redder, or a duplication greener.
So array CGH is far superior for detecting those microdeletions and microduplications and mosaicism, which were just invisible to traditional karyotyping. But what is the major structural anomaly that array CGH cannot detect?
Its main limitation is that it cannot detect balanced chromosome rearrangements.
Like translocations or inversions?
Exactly. Because in those cases, genetic material is just rearranged, but neither lost nor gained. So the DNA content remains 1-to-1. The array just sees a yellow signal across that region. It tells you nothing about the rearrangement, which may still carry significant clinical risk for that person's offspring.
And the power of these arrays has created a whole new set of clinical challenges related to interpretation, especially this high prevalence of variants of uncertain significance.
That's the core problem. The array is so sensitive it just picks up everything. First, you get CNVs of uncertain significance, or VUS, where you simply don't have enough clinical data to conclude if this change is pathogenic or benign.
So you should be cautious.
Clinicians have to adopt an innocent until proven guilty approach.
And then there are these CNVs that introduce a kind of gray area of risk, often called neurosusceptibility loci.
Yes, these are variants, often microduplications, that are associated with a low but real risk threshold for disorders like intellectual disability or autism. Whether the patient actually expresses the phenotype depends on other genetic or environmental factors. And it's incredibly hard to counsel families on a risk that might only be 10 or 20%.
And finally, there's the unavoidable issue of incidental findings.
A powerful array testing a child for intellectual disability might discover a microdeletion that happens to encompass the BRCA1 gene. Now, that finding is critical for the family's health, especially the mother's risk of cancer, but it is entirely secondary and often unwanted by the family in the context of their child's primary diagnosis.
Before we move on to the revolution of massive parallel sequencing, we have to acknowledge the technological bedrock that makes all modern genomics possible: PCR.
The polymerase chain reaction. It is the fundamental amplification engine of molecular biology. Invented in 1983, it uses thermal cycling, these short pieces of DNA called primers to define your target region, and a heat-stable polymerase to exponentially amplify targeted sections of DNA.
So it's a molecular copy machine.
It creates millions of copies of a specific segment, making it ample enough for analysis.
And PCR enabled the first major sequencing technology, Sanger sequencing, the method that delivered the Human Genome Project.
Right. Fred Sanger's method used fluorescently labeled chain-terminating nucleotides. These produced chains of differing lengths that were then separated by size, allowing a laser to read the sequence. It was a technological marvel, but as we said, scale was its limitation. $3 billion and over a decade for one genome.
And that leads us to the absolute game changer, next-generation sequencing, or NGS, also called multiple parallel sequencing. How did this drop the cost by a factor of 3 million?
It's all in the name. Parallel. NGS analyzes hundreds of thousands, sometimes millions, of DNA strands simultaneously. The cost reduction comes from that efficiency and scale, and it follows 5 basic principles that separate it from Sanger sequencing.
Okay, let's walk through the 5 steps of the NGS process.
First is library preparation. You take the DNA, you fragment it, and then you ligate custom adapter sequences to the ends. These adapters act like little anchor points. Second is amplification. These fragments are amplified to produce these dense clusters of DNA, and each cluster acts as a single separate sequencing reaction.
And if we're not sequencing the whole genome, we need a third step, which is capture.
Correct. If you are only interested in a panel of genes or the exome, you use a capture stage that physically pulls out only those desired fragments. Fourth is the sequencing itself. Each cluster is simultaneously sequenced via repeated chemical cycles, generating these short reads, typically 50 to 300 bases long.
And finally, the crucial step, interpretation.
That's alignment and variant identification. Specialized software takes those millions of short reads and maps them, a process we call alignment, to a reference template genome. And then any differences between the reads and the reference are identified as variants.
And how do they ensure data quality when you're dealing with millions of these tiny reads?
Through what we call depth or coverage. This is simply the number of reads that align at any given point in the genome. If you have low depth, you can't really trust the variant call. For diagnostic-grade results, a depth of 30 or more reads is generally accepted to confidently confirm a variant.
These NGS capture strategies define the scale of the investigation. Panels, whole exome sequencing, WES, and whole genome sequencing, WGS. What are the critical trade-offs between them?
Okay, so gene panels are the most restricted, they're cost-effective, and they provide very deep coverage, which increases your confidence in the result. They restrict the analysis to genes already known to cause a specific condition, which minimizes incidental findings.
But the downside is?
The downside is they only detect variation in known genes. If the cause is a new gene, you'll miss it completely.
So WES is the intermediate step.
Whole exome sequencing, WES, sequences all the coding genes, the exome, which is only about 1 to 2% of the whole genome. It's cheaper than WGS and usually provides deeper sequencing. It covers all the genes, regardless of suspicion, making it a great diagnostic tool. But it still risks incidental findings, and it misses variants in the non-coding regions.
And then WGS is the most comprehensive, yet surprisingly, it can be the most challenging to use clinically.
Whole genome sequencing WGS covers everything, coding and non-coding regions. It is the best for dosage abnormalities and for detecting variants in those tricky regulatory elements. But it's the most expensive, requires massive data storage, and because it detects millions of non-coding variants, it generates the most ambiguous results.
So the highest risk of incidental findings.
By far. And clinical WGS typically has shallower sequencing coverage than WES, which is another trade-off.
Looking beyond NGS, we're now seeing the emergence of third-generation sequencing. What's the massive advantage here?
These technologies, often called single-molecule sequencing in real time, or SMRT, offer significantly longer read lengths.
How long are we talking?
We're talking 10 to 15 kilobases, so 10 to 15,000 bases, compared to maybe 300 for current NGS. This is revolutionary because long reads can dredge these highly repetitive areas of the genome that just confuse the current short-read NGS methods.
And they're faster.
Faster, they use fewer reagents, and they are highly likely to supersede current NGS methods in the near future.
But regardless of the generation of technology, the clinical challenges of interpretation remain the same, especially around that variant of unknown significance, the VUS.
Oh, absolutely. The clinical workflow is constantly challenged by the sheer volume of data. We have complex algorithms to flag potential pathogenic variants, but if a VUS is detected, where there's just insufficient data to categorize it, we have to adopt that innocent until proven guilty approach. It requires expert genetic input in the context of the clinical presentation, which adds cost and complexity.
Let's look at how this technology is translating into real-world change. For rare neurodevelopmental disorders, NGS has been nothing short of transformative.
It has completely eliminated the diagnostic odyssey for so many families. Before NGS, clinicians had to analyze single genes sequentially, which meant they needed a strong, often very specific clinical diagnosis first.
A huge challenge.
It was. NGS allows for the simultaneous interrogation of multiple genes, sometimes thousands, and it's boosted the diagnostic yield in neurodevelopmental disorders from a historical low of maybe 10% to between 35% and 50% today.
And what does that actually mean for a family beyond just getting a name for the condition?
It provides clarity, it gives families prognostic information, it directs them toward support groups, and critically, it offers clarity for their future reproductive planning. It breaks that cycle of uncertainty.
When we pivot to common diseases like cardiovascular disease or diabetes, the challenge shifts dramatically from finding a single big error to understanding a polygenic picture.
Right. Most common disorders are polygenic. They are determined by these complex interactions between multiple genes and the environment. And so we study these not through sequencing one person, but through massive studies.
Like GOAs.
Genome-wide association studies, yes.
Yeah.
GOAs compare hundreds of thousands of SNPs between thousands of affected cases and healthy controls to find these common low-effect risk genes.
And this research is leading toward a really powerful preventative tool, the polygenic risk score, or PRS.
PRS are developed to stratify an individual's risk for a common disease based on the cumulative effect of hundreds or thousands of these low-risk SNPs. While they're not yet routine, integrating PRS will enable personalized management strategies in the near future. For instance, telling someone with a high polygenic risk for diabetes that they need to adopt aggressive preventative lifestyle changes decades before any symptoms would ever appear.
In obstetrics and prenatal testing, genomics has profoundly changed prenatal care, especially in reducing the reliance on invasive procedures like amniocentesis.
This is all thanks to non-invasive testing, or NIPT and IPD. It utilizes cell-free fetal DNA, or CFF DNA, that's found floating in the maternal circulation.
And this CFF DNA, it comes from the placenta.
It originates from the placental trophoblasts, yes. And it's robustly detectable as early as 9 weeks of gestation.
So NIPT is widely adopted now for screening for aneuploidies.
Right. NIPT uses NGS to sequence and quantify chromosome-specific sequences in that CFF DNA. It's highly accurate, around 98% for detecting trisomies 13, 18, and 21.
But it's not perfect.
We must always remember the limitations. False positives can occur because of something called confined placental mosaicism, and false negatives can occur if there's just too little CFF DNA in the sample.
And then non-invasive prenatal diagnosis, or NIPD, takes us a step further to identify specific single-gene defects.
NIPD is often used to look for specific variants that are paternally inherited or arose de novo and are not present in the maternal genome, like for achondroplasia, And crucially, research is advancing quickly to use NIPD for autosomal recessive conditions, like cystic fibrosis, by analyzing combinations of carrier variants from both parents at the same time.
And for families with known severe genetic risks, pre-implantation genetic diagnosis, or PGD, remains a very high-tech option.
PGD is an adjunct to IVF. It involves genetically testing a single cell from a developing embryo before it's implanted, allowing couples to select only those embryos that are free of a known familial condition. It's proactive prevention rather than reactive testing during a pregnancy.
Oncology is perhaps the most advanced area where genetics is actively directing patient management, not just predicting risk. The classic example of this is the use of PARP inhibitors in BRCA12-related cancers.
This shift from just informing screening to informing treatment is monumental. For decades, women with BRCA12-related breast or ovarian cancer receive standard chemotherapy. Now, genetic testing for that specific variant is mandatory before treatment is even initiated.
And why is the presence of a BRCA12 variant the key to successful treatment with these PARP inhibitors?
Okay, so tumors with a pathogenic BRCA12 variant already have compromised double-strand DNA break repair. That's what BRCA is responsible for. The PRAP inhibitor then comes in and it blocks the second key DNA repair pathway, the single-strand break repair pathway.
So you're knocking out both forms of DNA repair simultaneously, but only in the cancer cell.
Exactly. This is what we call synthetic lethality. Since the tumor already has a defect in double-strand repair, losing the second crucial pathway, single-strand repair, is catastrophic for the cell. It drives the tumor cell toward apoptosis, while often leaving normal cells relatively unharmed because they still have their functional BRCA pathway.
It's a perfect demonstration of exploiting a known genetic weakness in the tumor cell to achieve highly effective, targeted treatment with minimal systemic side effects.
It is.
We often focus on human genomics, but microbial genomics is undergoing the exact same revolution.
It is. The ability to sequence a microbial genome quickly, again, for under $100 today, allows microbiologists to rapidly and accurately identify the causative organism for an infection and determine its specific resistance profile.
So you get immediate, targeted treatment instead of just throwing broad-spectrum antibiotics at it.
Exactly. And beyond the immediate patient care, the broader application is vital for public health. Microbial genomics is crucial for surveillance, allowing us to track the spread of pathogens, identify new variants, and reduce transmission.
And for developing new drugs.
Most importantly, yes.
Huh.
Understanding the microbial genome will drive the development of new vaccines and new antibiotics, which is desperately needed as microbial resistance continues to rise.
The therapeutic side of genomics is really where the future lies, starting with pharmacogenomics.
Pharmacogenomics is personalized medicine based on your genetic determinants. It predicts how an individual will metabolize and respond to a drug. For decades, we've known that polymorphic variants in enzymes like CYP2D6 affect drug kinetics, causing some people to be rapid metabolizers and others to be slow metabolizers.
And that profoundly affects drug persistence and toxicity.
It does. So genetic screening can inform the exact dose required to achieve efficacy without toxicity.
Is there a classic example of this in practice?
A classic one is the enzyme thiopurine methyltransferase, or PPMT. Genetic screening for TPMT variants can identify patients who have reduced enzyme activity. If they're treated with standard doses of drugs like azathioprine, which is used for cancer and autoimmune disease, they accumulate toxic metabolites.
But with screening.
Screening allows clinicians to treat them successfully, but with much lower, safer doses.
The ultimate goal, of course, is gene therapy and gene editing, actually repairing or replacing the mutated genes themselves.
Traditional gene therapy, which often uses viral vectors to replace defective genes, has seen some success. But challenges around the immune response and the safety of the vector remain.
But the truly revolutionary development, the one that gets every geneticist excited, is CRISPR-Cas9.
CRISPR-Cas9 is the most accurate, efficient, and specific genome editing tool ever invented. It's essentially a pair of molecular scissors that are guided by a piece of RNA to cut DNA at a precise, custom location. The speed of its development has been staggering.
So what's the timeline looking like for clinical application?
Well, ex vivo correction, so editing cells in culture and then returning them to the body, like editing immune cells, that is likely to become commonplace relatively soon. However, in vivo correction editing within the body, correcting genes and nerve or muscle cells, is much further away.
Why is that so much harder?
It's a massive challenge of delivery. Getting the CRISPR machinery safely and specifically to every single affected cell in the body is a huge hurdle.
We also have induced pluripotent stem cells, or iPS cells, which are offering new models for disease study and future regenerative treatments.
iPS cells are a huge breakthrough. They are adult cells like skin cells that have been chemically induced or transdifferentiated to possess almost all the characteristics of embryonic stem cells.
So you can make patient-specific tissues.
Exactly. They hold huge potential for regenerative medicine by allowing us to create patient-specific tissue models of disease like heart or brain tissue for testing drugs or studying how the disease develops.
And finally, there's pathway medicine, which is about manipulating the consequences of the genetic variant, not the gene itself.
This requires a really deep understanding of the disease pathogenesis. You can take Marfan syndrome. The defect is in the protein fibrillin, but that defect leads to the overactive upregulation of something called TGF signaling in the vessel wall, which causes aortic dilatation. Well, researchers discovered that the common antihypertensive drug, losartan, acts as a partial antagonist of that TGF signaling. It's now being studied in clinical trials because it shows promise in preventing aortic dilatation.
So you're manipulating the biochemical pathway that was altered by the genetic defect, even if you can't fix the gene itself.
Precisely.
As this technology races forward, our ethical and clinical standards have to follow very closely behind. The sheer speed and accessibility of these technologies make their safe and ethical application absolutely critical.
The ethical implications are just profound because a genetic result, it's not just a piece of data. It is permanent, predictive information that impacts the entire family unit. It forces us to ask some really tough questions about who should be tested and when, especially when we're talking about testing healthy individuals for conditions that may never even materialize.
Let's use the scenario we discussed earlier to bring these concerns into sharp focus. We have a 32-year-old pregnant woman who has been referred for BRCA2 testing. The reason? Her healthy 65-year-old mother, who is of Ashkenazi Jewish ancestry, participated in a direct-to-consumer test.
The DCT.
A DCT. And she discovered she carried a pathogenic BRCA2 variant. Now, the patient herself has an identical twin sister, a 2-year-old daughter, and there's no significant cancer family history other than this one DCT result.
What a complicated situation. The DCT result itself is the starting gun. It forces this cascade of medical decisions that the family may not be prepared for at all. It concerns the patient, her sister, her daughter, and even her unborn fetus.
Okay, let's start with the fundamental ethical tenet that surrounds predictive testing. Testing an unaffected individual for a known familial variant, specifically as it regards children, like her 2-year-old daughter.
The clinical genetics community has a very clear, long-established rule here. Predictive genetic testing for adult-onset conditions should be avoided in childhood if no clinical benefit is accrued during childhood.
So since breast cancer screening doesn't start until much later in life, there's no benefit to knowing the BRCA2 status of that 2-year-old right now.
None whatsoever. And the purpose of that principle is to maintain the child's autonomy.
Her right to an open future.
Exactly. We have to preserve the child's right to decide for herself whether she wishes to undergo genetic testing when she is old enough to understand the profound implications of that result for her health, her career, her insurance, her own identity.
And this principle immediately extends to the pregnant patient's situation regarding her fetus.
Precisely. If the pregnancy is continuing, testing a fetus for an adult-onset condition like BRCA2 susceptibility, which can't be prevented or treated until adulthood, is highly controversial. The purpose shifts from health to, well, reproductive decision-making, which many would argue is an unacceptable burden of knowledge to force onto the child or the family without a clear, immediate clinical benefit.
And we can't forget that a genetic result is intrinsically a family result. It directly impacts the patient's identical twin sister.
Testing the patient for this known familial variant immediately reveals critical health information about her twin and other relatives. And this just highlights the absolute necessity of rigorous pre-test counseling. Before any serious health testing is undertaken, clinicians have to ensure the patient has considered the family-wide implications and, where possible, discussed the results with potentially affected relatives like her twin.
Finally, let's return to the source of the problem here, the direct-to-consumer test, the DCT. What are the significant drawbacks that cause this whole cascading clinical referral?
There are two massive issues. The first is the lack of professional support. The mother took the DCT, as she said, for fun. She completely bypassed the necessary face-to-face genetic counseling that must precede tests with serious health implications, like finding a cancer susceptibility gene. Counseling ensures true informed consent and preparedness for the result.
And the second drawback is related to the science itself and the potential for inaccuracy or, maybe worse, false assurance.
Inaccuracy is a huge problem. Now, DCT is excellent at identifying founder variants that are common in specific populations, like the specific Ashkenazi BRCA2 variant the mother carried. But many DCT packages rely on assessing overall risk using only a series of common SNPs.
And the problem is,
The problem is that only a minority of the true risk SNPs for most common diseases have been fully characterized. So the risk score that's generated can often be inaccurate.
so what's the consequence of that incomplete picture?
An individual might be falsely reassured that they are not at increased risk of a genetic condition because the DCT missed a critical rare variant, and that could result in inadequate surveillance or management. Or conversely, they might receive a terrifying result for a VUS that turns out to be completely benign upon clinical review.
So while it's empowering and accessible, DCT must always be approached with extreme caution, and any pathogenic result requires clinical confirmation and expert follow-up.
That cautious approach really defines modern clinical genomics. The sheer volume of information requires a framework of responsibility. An informed decision, made by a competent adult following experienced counseling, is paramount, given the permanence and family-wide consequences of this genetic knowledge.
So to synthesize what we've discussed, the technological speed in genomics is just breathtaking. I mean, moving from that $3 billion Sanger genome to the $1,000 NGS genome in a matter of decades.
And we've mapped the fundamental biology, from the double helix packaged in that silent heterochromatin all the way to the multiple proteins generated via alternative splicing.
And most critically, we are seeing these technologies fundamentally change clinical practice, shifting from purely diagnostic tools in rare disease to active, real-time treatment directors in oncology, perfectly exemplified by the ability of PRP inhibitors to exploit a tumor's existing BRCA12 defect.
The entire field is moving toward predictive, personalized medicine through tools like polygenic risk scores and, of course, the immense promise of gene editing via CRISPR-Cas9. However, the complexity inherent in interpreting CMDs, the profound permanence of the result, and the ethical responsibility we have concerning predictive testing in children mean that our ethical framework has to evolve just as quickly as our technology.
And that raises an important question for you, the listener, to think about. As the cost of sequencing falls further below that $1,000 threshold and access becomes nearly instantaneous via platforms like DCT, how will society balance the speed and accessibility of this powerful information with the critical requirement for robust, ethical, and expert clinical counseling?
That balance between accessibility and responsibility, that's what's going to define the genomic era.
It's a challenge of responsibility as much as it is a challenge of science.
Absolutely.
A truly deep dive into the blueprint of life and the future of medicine. Thank you for joining us, and we look forward to continuing the learning journey with you next time.